About the avid lure. So hybrid speciation, once thought to be uncommon, we now have many well-established examples of hybrid species systems, from Helianthus sunflowers to Heliconius butterflies to Italian sparrows. Today, I want to tell you about a punitive hybrid species system between the southern dogface and California dogface butterflies. This system was first proposed in 1914 by Gerold in a paper titled Species Building Through Hybridization. And this hypothesis is based off of some color pattern differences between the two species and intermediacy. The southern dogface differs from California in a few ways. First, melanization. The southern dogface, Zerina cisonia, has this black hindwing margin that's missing in the California. It also has a larger melanin pattern on the forewing than the California. And the California dogface has an iridescence that's just purple-blue that is completely lacking in the southern dogface. Now, this hypothesis of hybrid speciation has gone untested for over 100 years. Unknown to Gerold, though, at the time, there's also a color pattern which is UV. The southern dogface has a UV pattern on the hindwing, like melanin, that's missing in the California dogface. It also has a truncated UV coloration on the forewing compared to the California dogface. Where do we find these butterflies? The southern dogface actually ranges all the way from southern North America down to the tip of Argentina, while the California dogface is limited to only California and a little bit into Baja. Today I want to just focus on where the southern dogface meets up with the California dogface. This is a small part of the San Bernardino Mountains in Southern California, and this occurs for only about 3 months out of the year when the southern dogface populations boom and they migrate the range from Texas, they push through the Sonoran Desert and meet up with the California in the San Bernardino Mountains, where they supposedly hybridize and create an intermediate phenotype. So what is this intermediate phenotype? In 1906, W.D. Wright was the first one to actually describe these intermediates. And so he described it as a dogface that had a forewing of the California, a hindwing of the southern. And then there's a amount of variation that spans from very iridescent, very stereotypical California to completely lacking iridescence and showing more of the southern dogface. And because who doesn't love gentleman scientists, Wright would go on to describe some behavioral reasons why he thought they were hybridizing. And he states that as a lure, Eurydice is exceptionally energetic and persistent and not hesitant to ignore all rules of propriety of species and of genera. And he goes on to say that this possibility of a hybrid form will be a delightful study for some future student. And it only took 100 years for some future student to actually pick up this project. So the goals of this project was first to resample the San Bernardino Mountains. Do we even still find these intermediates 100 years later? And do these intermediates really reflect hybridization between two pure parental populations? We wanted to investigate the signs of intermediacy using 3 forms of evidence. First, wing morphology, using color pattern shape, then wing coloration, and then scale morphology. Then we wanted to look for genitalia evidence, and then lastly, a little bit of molecular evidence. So in 2013, right before I started graduate school, my advisor went to the San Bernardino Mountains and was able to successfully find these intermediates. 2 years later, I went back with him and we sampled across transects and we collected over 200 intermediate samples. But due to the time when we had to sample, we didn't actually find any southern dogface in the San Bernardino Mountains, but that could be an artifact of us sampling in August rather than the proper time frame in April. Classes. So for this study, we're using intermediates collected on the hybrid zone or proposed hybrid zone, and allopatric populations of California from more northern regions of California from the Ojai Mountains, and then the southern dogface, we're actually using Mississippi samples from basically our backyard. The first test I did was looking at color pattern shape using Image J and Morpho J for geometric morphometrics. And I picked 3 shapes to look at. First, the forewing UV. I didn't do hindwing UV because the intermediate and the California dogface completely lack hindwing UV. So I looked at UV pattern on the forewing, melanic pattern on the forewing, and melanic pattern on the hindwing. And this is discriminant analysis from Morpho-J, just establishing that all three of these color patterns are different between the southern dogface and the California dogface. And when we look at the intermediates with each species, we see overlap in every single color pattern, suggesting that there is intermediacy in these color patterns. Next, I wanted to look at actual coloration, and this was done by using oceanographic spectrometry to look at UV reflectance. And I used 4 regions across the wing. And here we can see that there are species-level differences in their color reflectance. The California dogface is the blue line, and the southern dogface is the red line. And you can see we're picking up around the 400 in the blue region, peak, which is probably this blue iridescence that is lacking in the southern dogface. And in this, we can see the intermediates actually have a color reflectance pretty much identical to the California dogface. Lastly, for this part, I did a series of SEMs and TEMs, which I'm not going to show today because they had no species-level differences in scales between southern and California dogface. It wasn't until I got access to laser microscopy to do 3D reconstructions, so I could actually see that in the UV reflecting regions of these butterflies, the southern dogface have flat scales, while the California dogface actually have curved scales, which we think may be the iridescence that we're able to see. So trying to get access to be able to do this for the entire set of intermediates to see if the ones that, to our eyes, don't have iridescence may lack these curved scales, but gotta get access first. We have another student in the lab who is looking at the genitalia, and he is doing the morphometrics for the genitalia of the males. And we have found that there is species-level differences in their genitalia. So we're going to also go through the hybrids or intermediates for that. Lastly, some molecular data. I sequenced 7 nuclear and mitochondrial genes across 6 individuals from each group. I picked these genes because they were recently used to reconstruct the hybrid phylogeny. So I was hoping that they would be diagnostically different between the 2 species. Turns out 4 of them weren't. No base pair differences between the 2 species. And for 2 other genes, it was a handful of differences. But for those 2 genes, we were able to see that the intermediate sequences were actually identical to the California dogface. They had no southern dogface. Lastly, I of course looked at CO1, and again we see that the intermediates all group with Iridis, the California dogface, and the sequences had absolutely no SNPs that were unique to the southern dogface. So to conclude all this evidence, we have sort of mixed results from the morphology in that the pattern shape suggests there could be intermediacy, the wing color suggests no intermediacy, and the molecular evidence suggests no intermediacy. With all of this, it's leading us to test an alternative hypothesis that this may not actually be hybridization at all. It may simply be phenotypic plasticity. In the southern dogface, they have a plastic trait where they go from yellow to pink, and they also lose their hindwing UV in between one form and the other. So it's very plausible that the California dogface may also be plastic, and that due to being in higher elevations from the San Bernardino Mountains, they may just be more melanized, and that's leading us to think that they're intermediate, but they're really just more melanized California dogface. So our future directions for this project, first we need to get more allopathic California samples. Our data set for the California samples are pretty low. We plan to go ahead with some genome sequencing. We intend to do RAD sequencing, but we've been talking lately about doing some low coverage full genome sequencing of a lot of individuals. And we would like to do some mate preference experiments. In the sister genus of these butterflies, Coleus, it's been shown that UV brightness is actually the #1 indicator of male mating success. And since these butterflies have such UV differences in both brightness and shape, we'd like to further test if that UV could be an isolating barrier between these 2 species. And at that point, I would like to acknowledge people, my advisors, some undergrads, former postdoc, fellow grad student, and the person who taught me all my microscopy skills. And of course, funding